I'm Karthik Shankar, and this week I'm going to be talking to you about species diversity. Why are we interested in diversity? Across the world, we get many different kinds of habitats and ecosystems which are very different from each other. So for example, we have forests, a variety of them across the world, which form a stark contrast to grasslands and savanna ecosystems. You have different types of forests like tropical forests and temperate forests, and for over 100 years, maybe even a couple of 100 years, people have recognized that these have very different types of species and very different numbers of species in them. For the longest time, it's been recognized that tropical systems, or what used to be called the torrid zones, have many more species than temperate, cooler areas. We can also contrast terrestrial and marine ecosystems. And in marine ecosystems, you have coral reef ecosystems, other benthic ecosystems, pelagic ecosystems, and so on. And each of these, again, is different in both the kind of species and the richness of species that they have. There's other kinds of comparisons that we make. There has long been interest in the differences between islands and the mainland in terms of, again, the nature of species as well as the number of species on them. There are many theories about why islands have certain species richness and composition, and also how different kinds of islands, continental islands and oceanic islands, might differ from each other. And specifically, the field, the domain that covers these questions, that is island biogeography, is something that we'll cover later in the course. But we can also be interested in the species richness of particular taxa. The most abundant single taxon in the world are beetles. As the biologist J.B.S. Haldane said, if there was a god, he had an inordinate fondness for beetles. We might be interested in the diversity and richness of fish, and within fish there are different kinds of groups that have very different levels of diversity. Bony fish versus sharks, for example. Similarly, if you look at reptiles, one of the groups of reptiles, namely the tuatara, has just one species, or maybe two, in New Zealand, whereas the group of that includes snakes, which actually evolved later than tuataras did. Tuataras probably evolved about 200 million years ago. Snakes evolved about 130 to 140 million years ago. And snakes have 3,000 to 4,000 species compared to tuataras. So these dramatic differences between taxa are also of interest to us. So while we use the terms species richness and diversity somewhat interchangeably, there can actually be multiple types of diversity. The one that we've been talking about is species diversity. How many different kinds of species are there, either in particular habitats or within particular taxa. But within this, we can also ask other kinds of questions. The one that we've asked so far is how many and what kinds. But we can also ask questions about structural diversity, such as the age and sex of animals, or the size and density of plants, or functional diversity, which is the role and function that these organisms play in ecosystems. And of course, at a finer scale, we can also examine diversity at the genetic level. Now, we're examining these in the context of communities. But what are communities? Communities comprise groups of species, but while we use the term very generically, the word community is generally used to denote a set of organisms in a particular place. The word guild is used to denote a set of organisms that belong to a particular trophic level or have a particular ecological role, such as carnivores or frugivores. the word taxa or taxon relates to the phylogenetic relatedness of that group. So very often we talk about, say, a frog community in the Western Ghats. Now this actually technically is an assemblage because it's a community of organisms that's in a particular place, but I'm also referring to a very specific taxon. And or we might refer to frugivores of the Western Ghats, and this is a combination of a guild because they're frugivores, but they're also in a particular place. And if I were to say frugivorous passerine and birds of the Western Ghats, that's actually that defines a taxon, a guild, and a community. Having said that, we typically use the word community more generically. It's just important to keep in mind that this can denote either place or ecological role or taxonomic group, or actually some combination or all of them. For the purpose of this lecture, we're also going to make certain assumptions. We're going to assume that all species are equal, whether it's a reticulated python or a tree frog, both of these incidentally from Great Nicobar Island. As far as our assessment of these is concerned, whether it's a large frog or a small frog, large snake, small snake, the group that we're assessing, we're considering species within that group to be equal in our measurement of species richness or diversity. We're also assuming all individuals to be equal. We know both of these not to be true ecologically. Nevertheless, for the estimates that we'll be using, we're making these assumptions. We're also assuming that species abundance is recorded using appropriate and comparable units. And this is a theme that we shall return to at the end of the talk. Before we get into measuring diversity or richness, we also need to briefly examine scales of diversity. Now, broadly speaking, and most commonly used in ecological parlance, you'll hear the terms alpha, beta, and gamma. But these are also referred to as, there's also other terms that are commonly used, such as inventory diversity, which is the diversity of the place that has been measured, point diversity, which is diversity at a particular location, and critically, the idea of differentiation diversity, which is the diversity between two locations. Now, this has been categorized as two kinds of diversity. That is diversity at a given location, and this can be, as mentioned before, it's within sample, which is known as point diversity. It can be between samples, but within a habitat, between habitats at a particular location has often been referred to as alpha diversity. But alpha diversity is often just the term used for the smallest unit, sampling unit, that the sort of smallest scale at which we're trying to estimate our diversity, within which we believe composition to be somewhat homogeneous. At larger scales, where we're comparing alpha diversities or combining the alpha diversities, that's known as gamma. And the difference between these two, or the differentiation diversity, is known as beta. Now, there is in literature and in usage, there often is no hard and fast rule about how these alpha, beta, and gamma are used. And in fact, at the larger scale, such as between landscapes, delta and epsilon diversity, these are rarely used terms. The important thing to remember is that in any scale of measurement or comparison, a single habitat or a single area of interest is your alpha. The difference between this and a similar area of interest is your beta. That difference or differentiation diversity is your beta. And now when you combine those, you have your gamma diversity. So one can always think of scaling up as a combination of multiple alphas and the beta between them combining to produce a gamma diversity. There's other sorts of terminologies that have been used that sort of simplify this a little bit, so that you can think of it as point species richness or single sample unit richness, sample richness, large area species richness, and biogeographic province richness. Biogeographic provinces also have strong evolutionary biogeographic influences, and so these are areas that have been isolated for long periods of time and have a particular species composition or a species pool within them. That's also of interest to ecologists and evolutionary biologists. And of course, other terms like habitat species richness and assemblage species richness have also been used. In the literature, when you're reading papers, you might not necessarily find that the authors have specified which of these they've used, but it's usually apparent when you look at the methods at what scale they're sampling, also depending on what kind of questions they're trying to answer. So having made some preliminary comments about different kinds of diversity, some caveats about their measurement, some comments about the scale at which we're measuring them. Today, let's get down to the idea of species richness itself. And this really can be simplified to the question, how many species are there, right? How many species of frogs are there, or how many species of butterflies are there? And we can actually ask this question at a global scale, and many people have. Various different methods that we won't go into today have been used to try and estimate the number of species on Earth. And you'd imagine after all of these years of exploring the world, of a suite of mathematical and statistical techniques that are available to us, that we might have an idea. And many scientists think they do. A number of colleagues over the last, a number of ecologists over the last 40 to 50 years have made estimates in the range of about 1 to 10 million. For example, Mora and colleagues use methods to estimate that the number of species is 8.7 million. Costello et al. Estimate 5 plus or minus 3 million. Chapman 2009 estimates 11 million, all roughly in the same ballpark. However, there is also an estimate from Larson et al. Who estimate that there are 1 to 6 billion species on the planet. So as you can tell from this, nobody knows for sure. Most ecologists are probably putting their money on there being about 10 million species, or upwards of that, but some clearly think it's very much higher. Now, while we're measuring species richness, we have to keep some additional caveats and issues in mind. Firstly, we have to be sure that we have identified species accurately. It doesn't really matter if we know their Latin names or not. If there are new species whose names are not yet, who haven't been described yet, and whose names have not been allocated taxonomic description, that doesn't matter as much. However, we need to know that the individuals that we are collecting, measuring, observing, identifying are being accurately binned into different species. Some of this depends also on the species concepts that are used, and we've also gone over the idea of the different kinds of species concepts that there are, and the debates and discussion around that topic as well. Obviously, a lot of it depends on sampling, and this is something that we'll address very specifically. The number of species that you encounter can change over space, and this is in fact often one of the questions that we're trying to address, but they can also change with time and season. And so the documentation that we do make of species richness, we do need to take into considerations issues of time, seasonality, and the spaces in which we're sampling them as well. A critical issue that we deal with, again, in measuring species richness is relative abundance and evenness, and this is something that we target very specifically when we talk about measures of diversity. However, when our interest is primarily in estimating species richness, the relative abundance of species, the fact that some species are common and others are rare, that many communities are not evenly distributed, does play an important role in our ability to estimate species richness accurately. And this, of course, relates directly to the idea of detectability. Some species are less detectable than others for the simple reason that they are less common, that they are rare, as just mentioned. But species may be less detectable than others for a number of other reasons as well. They may be more shy, less observable in some ways, or depending on the method that we're using, they may be less susceptible to capture. And so overall, they're less detectable to us, even if they're actually not more rare in nature. So how do we then go about actually estimating species richness? There's actually three ways in which we do this. One is by extrapolating species accumulation or species area curves. The second is by the shape of the species abundance distributions. And then the third is by a family of what are called non-parametric estimators. Let's go over each of these. So the first of these methods of estimating species richness is based on species accumulation curves. Now, in species accumulation curves, we basically plot the number of species versus effort. And this effort could take the form of the number of samples or individuals or time. Now, for example, if we were to plot effort on the x-axis and species on the y-axis, and let's say the effort in this particular instance is the number of species of trees that I find in a particular plot. So let's say I'm sampling trees in an evergreen forest in Kudremukh, and I lay 10 into 10 square meter plots, and I go and count the number of different species in each one. So let's say that I found 8 species in the first plot, I find only 7 in the second, I find 11 in the third, 5 in the fourth, some number 15, 14, and so on. Right? I'm finding different numbers of species on the different plots. Now, I could get more, I could get less. Now, this is not terribly helpful to me, right, to get an estimate of the total number of species in this particular area. Now, if I were instead to plot the cumulative number of species or the total number of species, then in the first plot, obviously, I still have just that number that I got originally. But now in the second plot, I have the number of species I found in plot 1, which is say 8, plus let's say out of the 7 species I counted in the first one, 3 are new, so I have 11 species. And then in the third plot, I have some more new species. In the fourth plot, I have some more new species. But as time goes by, as you can imagine, I'm going to find fewer and fewer and fewer new species, right? In fact, if I sample for long enough, I'm going to find that I find no new species at all. And if I plot this curve, this is going to plateau, and this helps me determine that there are, in fact, over time, no new species in the area that I'm sampling. This gives me confidence that I've sampled all of the species in this area. Now, as you can imagine, in reality, the species accumulation curve, of course, cannot go down, but it is going to be, it may not be as smooth as I've made it to look over here. Nevertheless, we can using a large number of samples. So for example, if we have 100 plots, we can ask the question, how many species would I have found in a single plot, and pick out one plot at a time and average those. Then I can pick out 2 plots at a time and average those, 3 plots at a time and average those. That gives me the same form of curve, but that's likely to be a smoother curve. Of course, we can get situations where the species accumulation curve does not plateau, and that's also of course possible. But when the species accumulation curve plateaus, we can use that to estimate the number of species. Species area curves are one form of species accumulation curve. When plotted for larger and larger parcels of land in the same region. So if I've got a broader area and I say how many species are found here and here and here, and I plot this along the same axis, that gives me a species area curve. The species accumulation curve can be used in 2 ways. As you'll see here, actually the jagged nature of the actual accumulation curve, because you may do many plots without actually finding new species, and then you find some more new species and some more new species. And of course, depending on the nature of the habitat, the heterogeneity in the habitat you're sampling, you may find a lot of new species at the beginning and it may just totally plateau out at the end. Or as you move from one part of that subdomain of that region to another, you might suddenly start finding another bunch of new species and not some for a while, and then you're sort of going to another slightly different area and you're finding new species and so on. Now if you go from left to right on this curve, you're deriving your estimate based on the accumulation, right? So this is we're using the species accumulation curve and its asymptote to derive an estimate of species richness. However, there are many cases where people have unequal sample sizes across these plots. So for example, if I've laid 4 plots and I have 100 samples in one, just 25 in another, and some intermediate number in the other 2, I might think to myself, maybe the plot with 100 individuals had more species simply because there were more individuals over there, and it wasn't that the particular area itself had supports more species. And so you can use the technique of rarefaction, where you go from right to left on this curve and you ask, how many species would there have been if there were only 25 individuals in my sample, which actually now has 100. So you can have you can take many samples or many sample subsets and say, I want to standardize this to the subset that has the fewest number of individuals, say 25, and I want to know how many species each of my other groups would have had if each one of those had only 25 samples in them, and that is done by a process, by a technique called rarefaction. The second class of methods to estimate species richness are parametric estimators. These use the shape of the species abundance distribution. We'll be talking extensively about relative abundance distributions, so I won't dwell on that here. The 2 major types of distributions that have been used are the log series and the log normal. The log series is non-asymptotic and uses this particular equation. The species richness is equal to alpha log 1 plus n by alpha, where alpha is the parameter that defines the log series curve and n is the number of individuals, and the log normal, which we'll revisit. Now the log normal can be truncated or skewed, which poses challenges in using it to estimate species richness, and the log normal also is sensitive to the types of abundance classes that we use when plotting the log normal. Park this for the moment. We shall be returning to both log series and log normal distributions when we talk at greater length about species diversity as well as relative abundance distributions themselves. This brings us to this class of approaches or estimators that are non-parametric. And they call it non-parametric because they are not derived from any parameters within the distribution or the relative abundance distribution itself. Now, there's a bunch of these. We'll go over Chow's estimators, abundance and incidence-based coverage estimators, and some based on resampling, such as jackknife and bootstrap estimators. There's several of these in the literature, and they use similar principles again based on the abundance distribution, based on the abundances of the species already sampled. But as I mentioned, they don't use a parameter that's derived from that distribution. What does that actually mean? The simplest of these is the Chow one, and the Chow one estimators, S Chow 1 is my now estimated number of species. This is what I'm trying to get at. Now, this is derived, this is obviously some number greater than the number of species I observed. So it's S obs plus something. Now, what is that something? That something is the number of species that I have missed, right? And this is derived from the number of singletons, that is the number of species that are represented by a single individual, and the number of F2 is the number of species that are represented by two individuals. Now, again, why does F1 squared by 2F2 provide this estimate? One way of thinking about this is, there we go again, F1 is to F2 as F0 is to F1. What are we trying to say here is that the probability of finding the first individual of a species is the same as the relationship of finding the second individual of that species. So now if I solve for F0, then I get F0 is F1 squared by F2, but F2 is represented by twice as many individuals, and therefore my estimate of F0, that's the number of F is the number of species in that place that has 0 individuals in my sample, is F1 squared by 2F2, and S obs plus F0, or S obs plus the number of missing individuals, gives me my estimate of the number of species in the area that I'm trying to get this estimate for. Similarly, you have another Chow estimator that is based on the relationship between the number of species in the sample, but here the number of species in a single sample, the species that are found only in a single sample, and the species that are found in two samples, right? So instead of saying a single individual and two individuals, I'm saying I've now laid a lot of plots. Now, how many species did I find that were there in just a single plot? I'm making the same comparison between Q0, Q1 and Q2 that I did between F0, F1 and F2. And I'm saying the number of species that are found in no plots, the relationship of that with Q1 is the same as the relationship of Q1 with Q2. And that gives me the number of missing species that I add to S obs to get my Chow estimator. This is useful in areas where, this is useful in cases where the number of individuals in my sample doesn't lend itself as nicely to the analysis as saying I found the species at I found a number of species only at a single location, and I found a number of other species at 2 locations or at 3 locations and so on. Along the same lines, there are also what are called abundance-based coverage estimators that use the relationship between abundance and abundant and rare species. Rare is those that are less than 10 individuals, abundant is those that are more than 10 individuals, F1 again is the number of species of a single individual, and so on. And we use these estimates, these to sort of get an estimate of the number of species that we have missed. how this is different is basically the principle over here is that I'm likely to have sampled all my abundant species. It's really the rare species that I've missed, and so I'm sort of fine-tuning the estimator to say amongst this rare species, how many have I missed? Like with chow1 and chow2, you can do this using abundances, and you can do this using incidence, similar to the abundance estimator, but based on the occurrence of species in samples. And broadly speaking, it's been found that Chow's estimators provide the minimum estimates of richness and assume homogeneity, and that's why the abundance and incidence-based sample estimators try and get at what proportion of rare species did I miss. There's also a class of estimators based on resampling. We won't go into the details of this. Jackknife estimators were first used in mark recapture. Again, similar to the Chow's estimators, Jackknife 1 takes into account the number of species in a single sample. Jackknife 2 takes into account the number of species in a single and two samples, and uses these to derive an estimate, use a resampling approach to derive the estimate of the number of species in the given area. All right. Yeah, this is the equation for Jackknife 1. As in all of the others, there's sorry about that. There's an observed number of species, and we're using a resampling approach here to determine the number of missing species. One of the key questions that we grapple with when we go into a study system with a question that is based on species richness. So we might be comparing the species richness of a forest and a grassland, as we started out with, or a temperate tropical forest, or more likely, very often, two forests adjacent to each other. Evergreen forest and a deciduous forest, or two forest patches such as, say, the forests of Mudumalai versus the forests of Anamalai, and so on, right? So we're going in with a question about which forest has more species of trees or more species of birds, because we have some hypothesis. Our hypothesis might be that the species that the sanctuary that has the larger area, or the region, the forests of Mudumalai are, say, smaller or larger than those of Anamalais, and so the larger area has more species, and that's our hypothesis. Or it could be that the ones at lower latitudes have higher species, the ones south of the Palghat Gap have more species, and so on. We can have any number of such hypotheses, but in order to test the hypothesis, we actually now need to get a precise estimate of the number of species. So when we start out, one of the questions that we're going to ask ourselves, and this is going to be a central question in quantitative thinking about ecology, is what's my study design, right? How am I going to get the best estimates to answer my ecological questions? With estimating species richness, we often have to think about what's a sampling rule. Very often people give rule of thumb or book answers like sample 10% of the area and things like that, or collect data from 30 plots. But as you would recall from the species accumulation curve, it's entirely possible that your area is very homogeneous, and by the time you've done 10 plots, or you've done 1% of the area, or 10% of several hundred square kilometers is just far too much to sample anyway, right? And you've done some sampling and you find that actually it's plateaued very nicely, right? Let me plateau it even better. On the other hand, there could be diverse areas with multiple habitats, and you're actually trying to get a sense of all of them, and you sample there for months and months and months, and you're doing a PhD, and your curve still looks like that, and you're looking at it thinking, I'm not sure I've captured all the species over there. And so you do need to have some stopping rules, one, of course, the one that I've demonstrated over here, is when the accumulation curve has reached an asymptote. Now this is not always possible. Another is to make species richness estimates using one of the parametric or non-parametric estimators, and when those estimates stabilize, even though the accumulation curve has not reached an asymptote, you believe that you have attained a fairly robust estimate of the number of species. Other stopping rules have been used, such as when all species have at least 2 individuals. This occurs only in fairly depopulated communities. There's as when we look at relative abundance distributions, we'll see that most communities have large numbers of rare species, and so we very rarely get to a point where when all species have two individuals in the sample. Similar to the resampling approach using bootstrapping and other procedures, we can also subdivide the sample and resample it in order to determine whether we've reached a reasonable approximation of the number of species present in the area. All right, to close things out, there's a few things to think about. Estimators may not always be comparable, right? So, in general, we need to think about which estimator we want to use, and to what extent we want an absolute estimate of species richness and a comparable estimate of species richness. So in that example of Mudumalai and Anamalais that I gave you, does it matter whether my estimate of species, let's say that I estimated 50 species for Mudumalai and 65 for Anamalais, and let's say that they both underestimates by a similar proportion. Does it matter that the actual values are 60 and 80, or does it only matter to me that they're relatively and proportionally the same, and allow me to derive those inferences about the ecological drivers of that difference? A more important, an equally important thing to think about is the precision of the estimate. Now, very often the estimate, or even your asymptote, will give you a particular value, 55, 141. What does that mean? Does the true value, do I know where it lies relative to the estimate I'm making? Are my confidence intervals, or is the potential variance of that estimate high or low? Based on the sampling methods, we can also arrive at confidence intervals for our estimates, and these are useful because we want to know that given the sampling that we have carried out, what is our confidence that the true value lies close to this estimate that we've derived, or that it would actually be quite variable, and so a variable estimate means that maybe my species richness estimate was 40, but maybe the true value lies between 10 and 100, and that's not terribly helpful for me if I'm making comparisons with another area or answering an ecological question. On the other hand, small variances and tight confidence intervals allow me to make those comparisons and ecological inferences with greater strength. Of course, there's always effects of sample size. We've talked about areas as if they were homogeneous, but even particular habitats. So I might be comparing a wet versus a dry habitat, but the dry itself might be heterogeneous, and heterogeneous in ways that we might not perceive. I might be confident that I've classified my forest into wet evergreen, moist deciduous, dry deciduous, scrub and thorn, or along elevational gradients, and whatever form of classification that I'm using. However, there might be heterogeneity within those areas that aren't necessarily apparent to us until we go out and measure those other things. So there's other forms of heterogeneity that we might not be capturing, there's different kinds of rarity. Again, this is something that we will return to, but as we've mentioned, the challenge in estimating and getting good estimates of species richness is not the common individuals, right? You go out into any part of the city or town, and you're going to encounter the 5 most common birds, in the first half an hour, even if it's not a particularly wooded area. Similarly, you can go out and sample fish in a pond, or frogs in a stream, whatever your taxon of choice. Butterflies, take a net and capture some insects. What you're going to get in the first minutes, hours, days, weeks of sampling is all of the common species. Our challenge really overall is to capture those rare ones, whether they're truly uncommon or just hard to detect, high in abundance but localized. So there's actually different forms of rarity, all of which makes that our particular challenge. Finally, there's software packages that calculate a suite of indices, and our brief advice on that is resist temptation, because it's really important for you to know which measure of species richness, which estimator best suits your data, why you're calculating that, and all of this goes back to the scientific or the ecological question that you have. So even though there's all of these available to you, they used to be available in software packages, today they're available in R. Resist the temptation to calculate all of them. Sample size, replication, study design and planning are absolutely critical to getting good estimates of species richness. Going into an area and saying, I'm going to sample as much as I can, or I'm going to sample, I'm going to freewheel through this, sample in some ad hoc manner, and some statistical method or some software will help me solve this later is a really, really bad idea. So there's no other way than to have good sample sizes and good replicates. Even though many of these estimators are available, often we know what species richness itself really means. It's just the number of different species, and we're always making an additional set of assumptions when we calculate estimators. So species richness, especially if it's based on a good study design, based on a sufficient number of replicates, based on an asymptotic species accumulation curve, actually serves the purpose really well. We will be talking about measures of heterogeneity such as Simpsons or alpha and various other types of indices in upcoming lectures. But as I said, when our questions in ecology are about the number of species, which itself comes with a certain amount of complexity, as we'll see, because these species we assumed here are equal to each other, but in some sense they're not. Even within a family of within a group of frogs, you have stream-dwelling frogs, tree-dwelling frogs, forest floor-dwelling frogs that belong to different families. There's already heterogeneity over there. There's already evolutionary history, and we're kind of lumping some of that for now when we're making inferences, and we can disaggregate those and look at them separately. But sort of in any group that we examine it for, we do make some of those assumptions of equality, and so one should always be more cautious about making additional assumptions when we derive estimates that we're going to use further downstream in our analysis of what causes those patterns in nature. All right, so that's it for now. We'll come back to species diversity in space and time in our next lecture.